Hello, everyone. I'm Laura Dormer from the Evidence Space, and today I'm delighted to be joined by Xabier García de Albéniz, who is Vice President of Science at Adigens Health. Welcome. Thank you for joining me. To start us off, can you tell us a little bit about yourself and your role at Adigens Health?
Thank you, Lara. Thank you for having me. I'm a medical oncologist by training. For the last 16 years, I've worked on epidemiology with a focus on causal inference methods. I've worked in academia for about 8 years, specifically in the program of causal inference from the Harvard T. Stein School of Public Health. And after that, I worked for about 8 years in a company where the main line of work was implementing post-authorization safety studies. And there is where I got exposed to a regulatory environment. And there I had to learn what would be the requirements by the European Medicines Agency or the US FDA when it comes to evaluating different safety studies. Recently, I've joined Adigens Health, which is a consulting company where we are very few people with deep expertise on our own areas, which means that we get very difficult questions from our clients, and that is what makes this work fun and very enjoyable.
It's interesting to hear that you've worked both as a clinician treating patients and as a methodologist, so designing studies. How has that combination shaped the way that you think about generating evidence that is both scientifically rigorous and clinically meaningful?
The evidence that we contribute to generate is evidence that is used to guide decisions on medical interventions for patients. And that has two implications. First, because we aim to guide decisions, we need to use a specific family of methods, which are causal inference methods. And that's one of the key areas of expertise in Adigens Health. Second, because those decisions are over medical interventions, we need clinical expertise. And we need that clinical expertise not only to identify which are the decisions that we need to inform, that sometimes come from the sponsors or the clients, but also to contribute to the design and on the implementation of the study. How does that work? For example, it's very important to understand what are the factors that doctors use to choose over the different treatment options for patients. Because at some point, we might need to use that information to make the two treatment groups that we are comparing more comparable. When we compare two different treatments in the real world, we need to be sure that we are comparing apples to apples. And the difference from the treatments that those different groups receive come from clinical judgment. So I'm not meaning that I'm able myself to identify those factors. I might be able to do that in some cancer studies, given my background. But I can have a fluent conversation with clinicians and guide them to inform the study design by identifying those factors. And also, of course, because of my medical training, discussions about clinical aspects are very fluent with clinicians, and the client usually welcomes that.
So real-world evidence is becoming an increasingly important tool for regulators and HTA bodies, but some studies are still challenged or rejected. In your experience, where do things commonly go wrong, and are those problems typically introduced earlier in the study design than people realize?
So, in my experience, things can go wrong mainly because of two reasons. And these two reasons are connected. The first reason is that the data might not be appropriate for the question that we're trying to answer. And there can be many reasons why the data might not be fit for your purpose. There can be measurement error, which means that whatever information is in your database is not represented the real life. There can be missing information back to the factors, the clinical factors that we discussed before, there might be a very important factor to decide one treatment or the other that is not present or captured in the data. Or even I might be planning to combine different sources of data that end up being very different. For example, imagine that I want to complement a randomized clinical trial or a single-arm clinical trial with an external control arm that comes from the real world. The data generation of these two data sources can be so different that you cannot combine them. So that's one of the reasons why things can go wrong. The second reason is that your study design might not be robust enough to avoid some type of biases, or your study design is giving you some effect estimates that is not conveying the full picture of the effect of treatment over time, or maybe you have not done your best to break your design and study results. And what do I mean by breaking your design and study results? Every study, both randomized clinical trial and real-world data studies, rely on some assumptions. Assumptions can be related to the causal identification of the effect. Those are called identifying assumptions. And one example is what I mentioned before, comparing apples to apples, being sure that the only difference between the two groups that I'm comparing is the treatment that I'm evaluating. That the technical term is exchangeability, and that's one example of an identifying assumptions. There are also assumptions related to the study design. A very common study design is a cohort design, where I have two treatment groups and I follow them over time. But there are other study designs. For example, instead of comparing two groups of people, I compare the same person to themselves, just by comparing two different periods of time. These two studies will have different assumptions related to transient effects, etc. And then, of course, there are assumptions about the statistical methods. Every model that we use have their own assumptions. How we model or categorize the variables also bring different assumptions. So whenever I implement a study, I need to try my best to break these assumptions and see whether my results are consistent to different study designs, to different statistical models, etc., and they can resist all these challenges. The reason why I mentioned at the beginning that the fitness for purpose of the data and the study design are connected is because you should do a feasibility evaluation of the data before you go ahead and analyze all those data. And to inform that feasibility evaluation, you need a fully fleshed study design, including all the sensitivity analysis that I discussed to try to break all those assumptions. So there is where the connection lies. First design, the feasibility evaluation, and latest, the study implementation.
If many of these problems originate at the design stage, what are some of the key decisions sponsors should be getting right before any data are analyzed?
So I think it's very important that first, the study sponsor should classify the study that they want to make in one of these three buckets: a descriptive study, a predictive study, or a causal inference study. And that's important because that will guide the type of methods that we need to design and implement the study, and that will guide how we interpret the number that we get at the end. So the first, classify your study into whether it's descriptive, predictive, or a causal inference study. Second, use the appropriate methodological framework for your study design. Because many of the interactions with regulatory bodies are about making clinical decisions. A lot of these studies are causal inference studies. So our recommendation here is that a good approach to design and implement these studies is using the target trial framework. The target trial framework works by specifying your research question in the form of a target trial, and then using real-world data to emulate each of the components of that target trial. Regulators like the EMA like the target trial framework because of its transparency, because it also facilitates the dialogue between trialists and other stakeholders in the study design, and of course because it avoids some types of bias. In my experience designing and implementing studies, one advantage of the target trial framework is that it helps a lot interacting with the clinicians. I mentioned at the beginning that it's very important to bring in clinical expertise into your study design and implementation. And clinicians are usually more comfortable discussing a randomized clinical trial because they have read thousands of clinical trials in their professional life than discussing a real-world data analysis. There, at least today, they are, I think, less used to those type of studies, but they are pretty comfortable discussing randomized clinical trials. Using the target trial framework, you can blend in very well the information that you gathered from them by discussing a hypothetical trial into the design of your observational study. Another advantage of the target trial framework is that it allows the sponsors to document what's the process that went from the original research question, that sometimes we call it the index trial, to the question that you are actually answering with your available data. That might be a different question because the available data will impose constraints of the type of study you can design and analyze. For example, based on the data that you have, you might need to adapt your study population because you're missing a variable to define eligibility criteria, or you might need to rethink your outcome because the outcome that you want to estimate is not capturing the data, but still the data has relevant information to study other outcomes that are still clinically relevant, etc. So the target trial framework, by doing first a full specification of your index trial, then a feasibility evaluation, and then the adaptation of the target trial, lets you convey to the regulator very transparently why you're doing what you are doing. And finally, another aspect that I think is worth mentioning is that the target trial framework works well in different settings. It works well in a purely real-world data analysis where you just use real-world data to compare two groups, but it also works very well when you want to combine clinical trial and real-world data. For example, because you want to use an external control arm. The idea of your target trial helps you in both cases to articulate very well your study design. So back to your question, what's a key decision here? A key decision is using an appropriate framework for design and analyze your study, and in Adigens we recommend to use the target trial framework.
One concept that often comes up in discussions around causal inference is time zero. For those who aren't methodological experts, what is time zero and why can getting it wrong undermine an otherwise well-executed study?
So time zero is very simple. Time zero is the time when we start counting the outcomes. Imagine that we are doing a study on survival. Time zero is when I start counting the deaths during the follow-up of my study. To avoid introducing some design-related biases, it's very important that time zero is aligned with the time when I apply the eligibility criteria and when the time when I assign patients to the different treatment strategies. If you think about a randomized clinical trial, you're probably thinking, well, of course, this happens in every randomized clinical trial. In a randomized clinical trial, I apply the eligibility criteria, select my eligible population, randomize them to treatment groups, and then start the follow-up there. That alignment is very clear in a randomized clinical trial. I would never do in a clinical trial, I would never start treatment before applying eligibility, right? That never happens because that would be a way to introduce selection bias if treatment can affect eligibility criteria. Or in a randomized clinical trial, I would never restrict my analysis to people who have survived for a period of time. So the same way that I would never do that in a randomized clinical trial, I should not do that in an observational study. It sounds easy, aligning these three components, but in real-world data, such alignment might not be straightforward. For example, imagine that I want to compare treatment versus no treatment. Time zero for my treated group is going to be very obvious. It's the time when they start treatment. But time zero for my untreated patients is not defined. If my study period is 10 years and I have people who have not been treated for 10 years, what's the time zero? Can be any day of those 10 years. So the target trial framework gives you resources to design a study and aligning properly time zero with eligibility and treatment assignment in this situation. Another situation where such alignment is not straightforward is on some type of research questions. For example, imagine that I want to compare different durations of the same treatment. And this is a question that is common, for example, in cancer. For how long should I give chemotherapy as an adjuvant treatment for people who had surgery for cancer? 3 months, 6 months, I don't know. So in a randomized clinical trial, it's easy to do. I randomize people to one arm or the other, and they receive the treatment that I am supposed to receive. But in observational data, where I don't have randomization, at baseline, at the time when they are eligible, everybody is starting chemotherapy. I don't know for how long they are going to receive it without looking into the future. And I'm saying that we should not look into the future, but because that would mean misalignment treatment with time zero. So there are situations where such alignment is not straightforward, but the target trial framework, again, gives you resources to deal with that successfully and avoid introducing design-related bias.
And finally, looking ahead, what's the one piece of advice that you would give sponsors who want to generate evidence that's credible, decision-ready, and able to withstand scrutiny?
So my one piece of advice would be investing on having access to expertise on causal inference and use that expertise not only to the analysis of your study, but from the inception of the research question. So make sure that it's really a causal question throughout the study design and, of course, at the implementation of the study. Causal inference methods have evolved substantially over the last 2 decades and are becoming mainstream in the last decade, I would say. They have demonstrated that they can contribute to avoid several design-related biases. They increase transparency, making the evaluation of the study easier, and the regulators are starting to recognize that. So I just want to finish by saying that using causal inference methods or the target trial framework doesn't mean that you're going to design a very complex study. You can, and there might be situations where you need to do a very complex study. But in my experience, in most of the times, you end up with a relatively straightforward study design that is free of some biases thanks to the use of the target trial framework, and that will fulfill the regulators' request successfully.
Xabier, thank you so much for joining me today and for sharing your expertise on this area. And thank you to everyone watching. I hope you found this discussion useful and gained some practical insights into what it takes to build real-world evidence that can withstand regulatory scrutiny. Thank you.
Thank you very much.